All right, so if everyone is here, I guess we can go ahead. So it's a pleasure for me to be here as well and to introduce the speakers today. So we're gonna have two speakers, and the main topic will be really mitochondria, as you know, was so nicely introduced before. But just not to interrupt our speakers any further, I'm just gonna go ahead and say a couple of words about them. So the first speaker is going to be Michiel. He completed his PhD in immunology at the Academic Medical Center in Amsterdam, and then he moved on to Utrecht, where he is right now. During his PhD, he studied how C-type lectins activate dendritic cells via interactions with viruses. And then later on, he studied inhibitory immune receptor signaling. And is actually what he focused on during his research that is now leading as a principal investigator in Utrecht. So today he's going to talk to us about some work published in, I think, December 2021, if I'm correct, in Neuron. And yes, I'm looking forward to hear more about it. The floor is yours.
Thank you, Miriam. Thanks for the introduction and thanks all for joining. I'll start sharing my screen so that you can see what I'm seeing. It comes through, right? Yeah, awesome. So today I'm going to present to you this paper. I'm probably have to go through quite fast, and I hope to have a lively discussion afterwards. If you want to be nasty, go ahead. I like fierce discussions, so bring it on. But before going into the data, I'd like to emphasize that this is a huge collaboration, which has been immensely fun to do. And this is a collaboration with Ramin Raouf, who did his PhD in this lab and now left for a postdoc in Amsterdam, and Hanneke Willema, who's luckily still in our department in my room. So those are my co-first authors, and both senior authors, also Linde Meijer and Niels Elkekamp, who had seminal contributions to this paper. So remember their names, remember their faces. These people are important for these data as well. So I'm going to start fairly simple, and that is that the immune responses come and go. So immune responses come when there's a pathogen or any reason for immune response to initiate. You get a peak of response and then they wane. This is all what we, I guess, expect and know. And together with the immune response, there's also a pain response. If you have a severe inflammation or a proper inflammation in your paw or hand or foot or wherever, anywhere, you'll notice that if you press this, it hurts. And also this pain response, or hyperalgesia, as usually it's more a oversensitized response to the normal pressure that doesn't induce pain. This you accept to be there if there's inflammation, but you expect it to leave the moment this inflammation is gone. And for me as an immunologist, I had to get used to this idea that there's neurons which are pretty long and that there's old stuff going on. So if there's an inflammation in the paw or your hand, this neurons get synthesized, the signal is routed through the nerve into the DRG, where the sensory neuron bodies are, cell bodies, and then rerouted through the central nervous system to interpret the pain. And about this first part, where the infection or the inflammation and the pain are initiated, there's pretty well known how this works. In the hand or paw, these macrophages or neutrophils that are there, they produce inflammatory mediators like IL-1, like prostaglandin, that sensitize neurons to become more sensitive and that you experience pain. But what is less well understood is how the resolution of inflammation or the resolution of inflammatory pain goes. And while you expect that this happens, right, the pain goes away when the inflammation is gone, this is not always the case. For example, in patients with rheumatoid arthritis or inflammatory bowel disease, at the moment we're pretty capable of suppressing the inflammation and to a stage that all inflammatory markers are pretty much down. But in some patients, or quite remarkable part of these patients, the inflammatory pain actually persists while the inflammation is gone. And to understand how this is possible, that this, why people progress into chronic pain and cannot resolve pain, the lab of Niels Elkekamp, or so our team in this case, studies this part of pain resolution. So in a model of transient pain, we study how pain resolves. And our part is there, how this inflammation and pain coexist. And in the model that we use, it's a fairly simple, I think, elegant model of carrageenan injection in the hind paw of a mouse. that, well, gives it transient pain. So what we do is we baseline measurements first in animals. We poke the paw and see when they retract. The harder you have to poke the paw, the less pain or less synthesized they are. Then we inject carrageenan and follow the pain response for a prolonged time. And this is a typical graph that you then get. So the open circles, I need to get my pointer. Oh, yes. So the open circles over there are the control sites. So we inject saline on one side, and you see that they don't respond. And the filled circles are the ones in which you get an inflammation, and indeed you get that these animals respond to a lower pressure, suggesting that they have pain, that they are sensitized to pressure. So this is the model I'm going to be using a lot in this talk. And what we found initially, and what we also knew from literature a bit, is that if you look in the dorsal root ganglion, so this area in the nervous system, or in the sensory nervous system, the peripheral nervous system, we see that in time there's an accumulation, but quite specifically at day 3, of macrophages, so F480 positive macrophages in the DRG, and this coincided with the resolution of inflammatory pain. So we asked the question, so what do these macrophages do in this, this process of resolution? So we depleted them, and we used a model in which the diphtheria toxin is only expressed in macrophages. Those are immune cells, for, I guess most of you will know. So if you inject diphtheria toxin in these animals, the macrophages and the monocytes will be depleted. And we do that from the start of the experiment throughout the experiment to keep them away. And when we do this, you see the same model. So pain is induced by carrageenan in control mice. This is nicely resolved within 4 days. But if you deplete macrophages, resolution completely fails. So this shows that you really need macrophages to resolve inflammatory pain. So this is right where we are. So you need macrophages. What I'm not going to show you, but what's in the paper, is that you need macrophages with an M2-like phenotype. So you need STAT6 or IL-4RA to be able to resolve inflammatory pain as a macrophage. And what's a key thing of M2-like macrophages, they express CD200 receptor. And what we found is that you need CD200 receptor on macrophages to be able to resolve inflammatory pain. I'm not going to show you the data, but I'll continue from this part. And what we find interesting, or what we found interesting about this, is that CD200 receptor expressed on macrophages, but on the other hand, on neurons, CD200, so the ligand, is expressed. And that got our attention because in 2016, it was published that if you add exogenous CD200 to a neuropathic pain model, the pain actually transiently leaves. So we thought perhaps CD200 on the neuron is needed as a ligand receptor interaction with macrophages. And that was quite a disappointment, because in all honesty, this was the first experiment that we did in this collaboration, and it showed no effect. So as you hopefully can see there in the black lines or black dots, it's the wild type animals, and in the bluish lines is the CD200 deficient animals, and these show the exact same pattern. They can just resolve inflammatory pain within 4 days, much as you would expect for wild type animals. So not CD200. So there is a second ligand for CD200 receptor, and this ligand was described to be only expressed in the gut. But luckily, we reached out to these people. So in Japan, Toshiyuki Koima, who was willing to share his mice with us. And we tested these animals, and indeed, this ligand for CD200 receptor was needed to resolve inflammatory pain. So not CD200 receptor DISEC. Again, here, nice induction of inflammatory pain, but then failure to resolve completely. So this makes the story as follows. To resolve inflammatory pain, you need macrophages with an M2-like phenotype. You need CD200 receptor on the macrophage, and as a ligand, ISAC1 on the neuron. Now, what's happening with these macrophages, right? Why did they go to the DRGs? Why did they go to the soma of these neurons during resolution? And this back in 2016 was a wild guess for us. but we decided to just try. And what we did is based on this paper published in Nature that showed that astrocytes can transfer mitochondria to neurons in the brain. We tried, well, why not? So for this, we used mice, which have Lisci mitochondrial flocks. That's their genotype, basically. So they express mitochondria 2 specifically in mitochondria in Lisci positive macrophages. And we just checked what's the signal that comes from neurons. And this is a graph or picture from this. What you see here in yellow is mitochondria, so that's the signal coming from the mitochondria that are at least processed or started in macrophages. WGA, which is a retrograde marker, so we injected that in the paw of an animal to see which neurons innervate the hindpaws, and then in this color, a marker for neuronal cells, from neurons. And what I hope you can appreciate is that in between all these cells, there's little dots, which are the macrophages that have infiltrated or that are in the DRG, but also we see staining in the neuronal cells or the neurons of these animals. And some of these neurons are yellow, so contain mitochondria, and they are WJ positive, so they innervate the hindpaw, but also some of them seem to not innervate the hindpaw. You can see here, you see these macrophages nicely close attached to these neurons in the DRG. And when we quantified this, I think this came out pretty nice. In naive animals, there's some background, some baseline signal coming from the neurons, but only at day 3, so after, at the onset of resolution of inflammatory pain, we see quite specifically at the site that was injected with lactic carrageenan, so that at the site of the inflammation, we saw an increase of signal of mitochondria coming from macrophages in neurons, and that disappeared at day 15. Now, to get somewhat more certainty of this data, we used another model. So we turned back to the depletion model, in which we just removed all macrophages from these animals and then injected at day 6 mitochondria, mitotracker deep red labeled macrophages. And in this experiment, we took some more controls along, namely animals that didn't have injected carrageenan, so no pain, or wild type animals. And I'll take you through this. So first, this is baseline. So this is depleted animals that got macrophages that are labeled but didn't get any pain. And they get this signal, so background signal, which is about the same as what we see if you inject PBS, so no label at all. Now, if you inject macrophages that are labeled in animals that have pain, you see an increase of this signal coming from the neurons, suggesting that macrophages transfer this dye, at least, to the neuron. Now, this doesn't happen if you sonicate these macrophages. So at the moment, these macrophages are just, well, broken down into pieces. There's no accumulation of dye. And if you inject intact macrophages in animals that did resolve inflammatory pain because they're wild type, they're sufficient for their macrophage pool, we also do not see an increase in the accumulation of this dye in neurons. So I guess together, or to me together, this shows that macrophages go to the DRG, where the cell bodies are of these neurons, and they transfer at least the dye to these neurons during the resolution of inflammatory pain. Now, in this paper, I mentioned earlier that in 2016 Nature paper about astrocytes, they showed that this is actually vesicles can do this. So we decided to test this as well. So we took, in the same model, we took the supernatant, spun it down to the supernatant again, isolated vesicles from that supernatant and injected that intrathecally, so in the back of a mouse. And what you can see is that only in intact vesicles, in the black lines, you see restoration of pain, so pain goes away, be it transiently, but not if you sonicate these vesicles. So you need intact vesicles and not components of these vesicles that can do this. So that's all nice, but it doesn't show necessarily that there is mitochondria that do this. So we used FACS to look at these vesicles, and we found at least that there's mitochondria staining in these vesicles, and also in the supernatant of Mitochondria 2 positive macrophages, there's vesicles that are mitochondria 2 positive. So the next step was to see if we could intoxicate these mitochondria, to poison these mitochondria, to see if we block the respiration, if they then could still resolve pain. And for this, we had to do a little trick to get enough mitochondria. So we cultured macrophages, lysed them and isolated mitochondria, or just before lysing them, we added mixothiazole, which blocks complex 3 and thereby blocks respiration. And here again in the black bars, these are the intact mitochondria injected, able to resolve inflammatory pain. And in the bluish dots, you see the mixothiazole treated mitochondria that cannot respirate properly and hence cannot resolve inflammatory pain. So now we're in this stage. You need macrophages to resolve pain. They need CD200 receptor, they need ISAC, and they need to be able to transfer mitochondria. So can we tie this together? Do you need CD200 receptor to be able to transfer mitochondria to neurons? And we turn back to the model that I've been using before, except for now in CD200 deficient mice. I didn't show you the data, but these CD200 receptor deficient mice cannot resolve inflammatory pain, so they they have persistent pain until days and weeks after injection of carrageenan. And if we inject in these animals mitotracker labeled macrophages, wild-type macrophages, you see accumulation of the dye in the neurons. And when you do the same thing with receptor deficient macrophages, you see that this accumulation doesn't take place, suggesting that you really need CD200 receptor to be able to transfer mitochondria to neurons. And the same thing holds true for vesicles. If you isolate vesicles again from wild-type supernatant cultures, you see resolution of pain. But if you do this, so if you isolate vesicles from the receptor knockout cultures, nothing happens. Showing that also on these vesicles, you need CD200 receptor to be able to resolve pain. So this is the summary this far, and if you've seen the paper, you've seen this picture. So what I think we've shown is that during pain, inflammatory pain, you need macrophages that go into the DRG to transfer mitochondria, either via vesicles or cell-cell contact. We cannot exclude that. But you need CD200 receptor, you need ISAC to restore inflammation. On the other hand, if you lack either of these molecules that I just mentioned, transfer doesn't happen and resolution of inflammatory pain cannot take place. Now, what to me, at least conceptually, is the nicest part of data is that this whole process seems to be independent of resolution of inflammation. I'll show you this. So I hope you remember this slide from the beginning of my talk, that sometimes in some patients, if you treat them, you're able to resolve inflammation, but inflammation keeps going, inflammatory pain keeps persisting. And this, and this is actually what we saw in CD200 receptor deficient animals. So we checked expression of cytokines, so expression of inflammatory markers in the paw of these animals, because we thought that CD200 receptor, being an inhibitory receptor, might prevent resolution of inflammation and thereby preventing resolution of pain. But we found that CD200 receptor and wild-type animals behaved exactly the same in their inflammatory parameters. So IL-1 production is the same in these animals and completely gone at day 7. So that suggested that at this stage, if there's no macrophages present that can resolve pain, so this suggested that at this stage, inflammation has gone, but if there's no macrophage present, pain persists. So that would mean that if we would add a immunosuppressant at this stage in macrophage depleted animals, that wouldn't help to resolve inflammatory pain. So we did this. So we used our diphtheria toxin model, again, depleting the macrophages, inducing pain, and either at the beginning of the experiment or at the end of the experiment, inject dexamethasone. Now first, I'll show you the data of the beginning of the experiment. Because that shows that if you inject dexamethasone, you can actually prevent induction of inflammatory pain, which makes total sense. So this is vehicle control with, so for the vehicle control for dexamethasone, which means you can just induce inflammatory pain, which persists much as expected. And if you add dexamethasone in the animals just prior and during the beginning of this inflammatory pain, well, there's no inflammatory pain, showing that dexamethasone can suppress this. Now, if you do this at day 6, in which wild-type animals normally would have resolved inflammatory pain and inflammation has gone, but not if you deplete macrophages, so then the pain persists. If you then inject dexamethasone, you see that this doesn't do anything for the inflammatory pain. And to me, this suggests at least that at this stage, while you need resolution of inflammation to resolve pain, resolution of pain itself is an active process that follows or is parallel to the resolution of inflammation, which is, to me, I think that's pretty cool. Yes. I'm at the end of my presentation. I want to thank you all for your attention, but again, I want to mention Ramin and Hanneke for their, well, for being them. They're awesome. And I want to thank Toshiyuki Koyama for sharing their mice, and Will for his help with all this. And I'd like to thank our funders to, well, they made this possible. If you have any questions, please ask. I'd like to do the best.
Thank you. Thank you very much. That was a great talk. So just to remind everyone, you're welcome to write down your questions into the chat box. You're also welcome to just raise your hand and unmute yourself if you want to have a direct conversation with the speaker. So we already have one question in the chat box, so I will start with that. More than one, actually, they're coming down now. Okay, so let's start from the first one. I will read a couple. We only have 5 minutes for the questions, but feel free, of course, to reach out to the speaker if I don't get to all the questions today. So the first question from Carlos Humberto Martinez Alvarez is, are these macrophage neuron relationships restricted to the DRG? Could this happen at the level of peripheral nerve terminals in the inflamed tissue?
Yeah, it's an awesome question. And the answer, I think yes. I would say this could happen in the periphery as well, but we didn't check yet. So we see it in the DRG. But I'm pretty sure that if we would isolate the sciatic nerve or look in the paw, we should see interactions there as well. But I have no data to confirm that, but I expect yes.
All right. So next question. Nice talk and interesting story. Naive question about why macrophage will transfer mitochondria to the neurons. Are mitochondria and the neurons unable to function properly?
Yeah, that's a interesting question as well. We philosophized, or I don't know if that's the right word in English, but we hypothesized that it's because during pain or during inflammatory pain, these neurons are under stress in such a way that their mitochondrial pool is just stressed, and then they would need help from macrophages to replenish this pool. And then the argument would be, or is, that neurons are pretty, expensive cells. You cannot really make new ones, and macrophages are, well, they're just replaced in a second. So it wouldn't matter if you would sacrifice 1 or 2 macrophages to save 1 neuron. That's what we think at the moment. Sounds good.
That question was from Chu-Wen Chen. And then we have a technical question from Luca De Feo. So he's asking, great talk, first of all, and were the CD200 receptor knockout mice conditional ones or constitutive?
No, they're full knockouts. So they're always knockout.
Okay.
If you have follow-up question, then I'm interested in why you ask.
Yeah, so yes, definitely. Absolutely get in touch if you have more follow-up questions. So from Estefania Nova Lamperti, she also says, great talk. Is the mitochondrial transfer exclusive to neurons, or do they transfer to other cell types as well? And also, what happens to the macrophage function after they release the mitochondria? Do you know anything about that?
Yeah, so I would, I've checked some other cells, so also some tumor cells get mitochondria from macrophages, but in vivo or in this system, I'm not sure. What happens to the macrophage after releasing mitochondria? Fair question, we haven't checked. So I hope there's multiple options, right? Either they can just make new ones and they live happily ever after. Probably not. I presume they either migrate away from the DRG and die somewhere. And in the worst case scenario, and I think that's also shown in prolonged pain models, is that these macrophages in the DRG gradually shift from an M2 phenotype to an M1 phenotype. And M1 macrophages actually are pain inducing. So that would also perhaps explain why, if you are not soon enough interfering with these processes, you get prolonged inflammatory pain or chronic pain.
All right. Then we have a few more questions. I'll try to get to all of them before the end of our time. So the next question is from Trent Evans. So he's asking whether there's evidence yet for improved or restored overall mitochondrial function in neurons that have received mitos from macrophages. And also he's asking if you know whether in the recipient neurons you have an idea of like the percentage of mitochondria that came from the macrophages.
Yeah, so the first question, do you know what happens to the neurons after they got mitochondria? So we tried to address this by looking in CD200 receptor deficient animals, so they do not resolve pain and they cannot get mitochondria. But quite to our surprise, we see that their oxygen consumption of the neurons actually normalizes. So although the pain's still there, their oxygen consumption is the same. Which I guess begs the question, what else? Is it just the DRG where we've been looking and we should look more in peripheral side of the neurons? Is it other functions of neurons like calcium buffering or redox balances? I don't know. We have to check, and that's up to the funders that I hope will fund our proposal to do this. But at the moment, I don't know. There was a second part of this question I forgot.
Yes, if you have any idea about the percentage of the
mitochondria. No, that's really, no, I don't know.
Okay. Okay, I will ask one last quick question, and then I think we might have to move on. So from Brian Campos Salazar, great talk, amazing data. How much of the pain resolution process can we attribute to an imbalance of the oxphosphate in DRG neurons?
Can you repeat that?
Sure. How much of the pain resolution process can we attribute to an imbalance of oxphos in DRG neurons?
As opposed to resolution of inflammation, I would say. I don't know. So what we see and what I always remain to feel intrigued by is that in CD200 receptor deficient animals, you see that the pain resolution initiates but never never completely finishes. So like halfway these of the resolution, they stop and they just keep on persisting pain. So if that is the initial resolution is because of the going away of the inflammatory mediators, and the second part, which fails, is because of the transfer of mitochondria, then I would say 50%. But that's, yeah, and that's not definitive data. That's just based on the data we have and speculation.
Okay, so there's a lot of more questions, but we might have to move on right now to the next talk.